Yeah, so hello everyone. So I will present you parts of my PhD results about lambda's mutagenesis, especially the estimation of lambda's mutation rates. So yeah, what is known about bacteriophages mutation rate is very scarce, but we do know that there is a constant rate of spontaneous mutations in DNA-based microbes. This is John Drake's work in the 90s. So we found that the rate of microbes, not just phages, but also bacteria, is identical at around 0.0033 mutation per genome. So interestingly, it is independent of the genome size and the lifestyle. And the implication is that the larger is the genome, the lower is the mutation rate. This is something you see here with the hosts that have a large genome but a smaller mutation rate. And here, the phages that infect these hosts that have a higher mutation rate. For single-stranded DNA and RNA phages, we have clues about why this mutation rate is this high. It is not just about the rule, but often they encode their own polymerase, more error-prone, and also they lack access to double-stranded DNA repair systems. But for double-stranded DNA phages that use the host replication machinery like lambda, we don't know why the mutation rate is this high. And so about the question, revising the mutation rate of lambda. So lambda is a double-stranded DNA virus that use the host replicative DNA polymerase 3. But it has a mutation rate that was estimated to be 400-fold higher than the hosts. But it was estimated with the fluctuation assay experiment a while ago. And this experiment gives a mutation rate per gene, so using a selection system on our target locus. And to compare this rate of lambda to that of the host, we need to use unverified hypothesis to convert the rate to a rate per base replicated. But the most concerning issue about this experiment is the fact that there is no published protocol or raw data to verify this mutation rate. So the objective was to obtain a more reliable estimate of lambda's mutation rate. And for that, we used three independent experiments. So two genomic experiments, the duplex sequencing and mutation accumulation, and one genetic experiment with the fluctuation assay. So for the genomic experiments, we used the mutation accumulation associated with the whole genome sequencing. So for that, the aim is to accumulate mutations in a lot of amplification cycles, so that mutation will accumulate across replications of the phage. And at the end, we will have a population that will be composed of mutated phage compared to the ancestral line. And for that, we did 150 transfers and 30 parallel lines that were originating from the same ancestral line to accumulate about 3 mutations per line. This experiment allowed the identification of mutations that are present in most genomes at the end of the experiment. The other approach is the duplex sequencing experiment, where there is just one single amplification step, so that mutation will be present in very low frequency. And to identify these mutations that might be after sequencing with the same frequency than replication and sequencing errors, We use the duplex sequencing approach, where after a DNA sharing, we added unique molecular identifiers at the end of each fragment and generated by PCR a lot of of duplications, so that after sequencing, we were able to reconstruct families of reads that share the same identifier and we hope the same sequence. So with these families, we were able to construct consensus sequences. So one consensus sequences will represent one strand of the original DNA fragment. And with this consensus, we were able to remove most of sequencing errors that might be just some reads of a family. But to remove PCR errors that might be in most reads of a family, we constructed a second consensus sequence using the base complementarity of the two strands of the original DNA, so we can only keep the true mutations. So with this experiment, we were able to identify the low frequency mutations that were accumulated in a single amplification step. But in this experiment, we did an underestimation of the number of DNA replications. And this is because we assumed that lambda was only doing the bidirectional replication like the host, which is not the case. Lambda also does the Welling circle replication. We also say that the phage infects one cell immediately after lysis, which is not really the case, and that one phage will absorb per bacteria. So all genomic experiments that I presented for the duplex sequencing and mutation accumulation define an upper bound of the mutation rate. But even though we were able to obtain a mutation rate that is close to the estimate that was obtained in the past, about 8-fold lower estimate. And interestingly, that was similar between the two independent experiments, the two independent approach to duplex sequencing and mutation accumulation, with a rate that is around 1 times 10⁻8 per base replicated. But these are upper bounds of the mutation rate. So to be sure that we have a mutation rate that is reliable, we use the fluctuation assay and we use the C2 selection system. So the C2 selection system is using the genetic switch of lambda. So simple. When there is expression of C1, there is a lysogenic cycle. When there is expression of CRO, there is a lytic cycle. There is also a protein that can be expressed at the same time as CRO, which is C2, and C2 will repress the lytic cycle and induce the lysogenic cycle by expression of C1. But a host produces a protease, FTSH, that will cleave the protein C2. So we are using a system where the host does not have FTSH. And in this system, C2 will accumulate and will force the phage in the lysogenic cycle, except if the C2 is mutated. So we use this system to do the fluctuation assay with C2 as a target. So the fluctuation assay has a simple protocol. First, we distribute a dilution of phage stock in a lot of parallel cultures of wild-type hosts. Then after 1 hour of amplification, so corresponding approximately to 1 burst of phage, we plate the whole culture of phage with the selective strain of Escherichia coli. And then we sequence the C2 sequence after PCR. But with this experiment, we are limited about determining the number of mutations that leads to the mutant phenotype that is called the target size. And for estimating the target size, we were confronted to two polyen in the C2 sequence, one polygene, one poly A, where there is a lot of insertion and deletions that accumulate in these two regions. So we need to remove those. But the issue is that C2, almost all positions in C2, when they are mutated, that will lead to the mutant phenotype. So we need to remove the polygene and poly A without disturbing the amino acid sequence of the protein. So for that, we used CRISPR-Cas system to edit the C2 sequence and to replace one G by an A without changing the amino acid sequence. And with that, we were able to obtain a mutation rate that was very close to what was obtained with the genomic experiments at around 2 × 10⁻8 per base replicated with the wild type phage, with a lot of insertion and deletions. So when we move in the influence of the poly-G, we were able to obtain a mutation rate that is closer to 5 × 10⁻9 per base replicated, very close still to that was obtained with the genomic experiments. And interestingly, the base pair substitution rate, so when we forget about the insertion and deletion, the base pair substitution rate is very similar between the wild type and the mutated phage. So it gives reliability about these results. And so the mutation rate of lambda is actually 16-fold lower than the previous estimate, and around 5 × 10⁻9 per base replicated. So as a general conclusion, we obtained similar estimates with 3 independent approaches. And we did assumptions in both approaches. The bidirectional replication for mutation accumulation and duplex sequencing. So that gives an upper bound of the mutation rate, and the removal of a poly-G for the estimation of the target size decrease the mutation rate. So we have a lower bound of the mutation rate with this experiment. So with that, we can say that lambda's mutation rate between 0.5 and 1 × 10⁻8 per base replicated, which is only 21-fold higher than that of the host compared to the 400-fold higher mutation rate that was previously estimated. And so it departs from Drake's rule that was estimated. So with that, I want to thank everyone that was involved in this project, especially my supervisors, Marianne and Naina, and you for your attention.